What I'm going to do is I'm going to tell you about the work that we've been doing recently on this multidrug resistant and globally disseminated clone of E. coli. I'm going to give you some insight into some genomics that we've done. I'm also going to tell you about a high-throughput genetic screen that we've done, and I'll show you some really nice and exciting data that we've obtained from those methods. And then I'll give you just a bit of a snapshot of a novel type of therapeutic that we've been able to work with and develop for treating UTIs. So just a little bit of background about these infections. I'm not going to spend too much time on background, but I just want to put the disease into some context. Urinary tract infection, or UTI, is extremely common, particularly among women and the elderly. Okay, so it's estimated around 50% or more of all women will experience at least one UTI in their lifetime. And these are essentially ascending infections, so the bacteria originate from the colon, then colonize the urethra, the bladder. They can make their way up the ureters to the kidney, and in some cases they can also transgress into the bloodstream and cause urosepsis. So these are really significant infections with regard to human health and healthcare facilities. They account for around 10 million doctor visits and around $1.5 billion in expenses every year in the US. As I've indicated, the majority of cases occur in women, and we're now seeing the emergence of an increased frequency of these infections being caused by multidrug resistant pathogens, hence the need to try and develop new approaches to treat these infections. So this is the major cause of UTI. Uropathogenic E. coli, or UPEC as I'll refer to it. And it has a number of virulence factors that enable it to colonize the urinary tract and cause disease. And I don't have time to go into a lot of detail about these, but I'll just mention a few. The capsule and O antigen, these are polysaccharide matrix or material that surrounds the entire bacterial cell and provides protection against the immune system. Many UPEC strains secrete toxins that damage epithelial cells. They also secrete iron acquisition molecules called siderophores that enable them to scavenge iron. And remember that the urinary tract is a nutrient-deficient environment. And they also produce a number of different adhesins on their surface. And these are really critical to colonization, because you can imagine if the bacteria don't stick to the epithelial cell lining, then they're washed away by the flushing action of urine. So over the last couple of years, my group has really focused on the study of a particular clone of E. coli called sequence type 131 or ST131. And we've defined this as the new global face of UPEC. So since 2008, we've seen the simultaneous spread and high prevalence of this clone across the globe. This clone is generally multi-drug resistant. It was first identified due to its association with an extended spectrum beta-lactamase, most commonly this CTXN15 type, which mediates resistance to oxaminocephalosporins and monobactams. And many of the ST131 strains are also co-resistant to aminoglycosides and fluoroquinolones. So there's not much in the way of antibiotics and the armory of antibiotics that we have that are able to treat infections caused by this clone of E. coli. They cause both, of course, community and hospital-associated infections, UTI and urosepsis, and they have a high number of virulence factors, so those factors that I just pointed out to you in the slide previous. So just recently we've completed a global phylogeny study of E. coli ST131 together with Scott Beatson and members of his group, and we published that in PNAS earlier this year. And in this study, we took 95 strains of E. coli ST131 from 6 different geographical regions, spanning 2000 to 2009. We generated a draft genome sequence of these strains. We assembled them, and we looked at the phylogeny by constructing a SNP tree. And what we identified were 3 major clades of ST131, as I've indicated here. And you can see that they cluster separately from other uropathogenic E. coli strains. And so they do have some differences in this respect. If I expand on this group of clade here, then you can see that it's quite complex, and I just don't have time to go into too many details. I'll just point out to you the major things. Firstly, you can see that the dominant group is this green group here that we've referred to as clade C. So all of the strains within clade C are very closely related. This clade C that you can see contains E. coli ST131 strains that have been isolated from all across the globe, so all 6 different regions that we sampled. They are all resistant to fluoroquinolone, and they all have exactly the same chromosomal mutations that mediate that resistance to fluoroquinolone. And so, in a nutshell, all of this data that we've got on this clade structure here suggests that what we've seen is the rapid emergence and global spread of E. coli ST131 from a single progenitor strain, which is really not what we expected to see when we first started out on this analysis. So we've started now asking questions with regard to ST131 and why it's been so successful. So is it due to antibiotic resistance? Well, certainly antibiotic resistance has played a major part. There are others in the field who have suggested that ST131 may contain certain metabolic factors that enhance its growth. It may survive better in the intestine. And most recently, we've looked at the ability of this organism to survive in the bloodstream, because we hypothesized it might have properties that enhance this or survival in this environment. So what I'm going to tell you about is some work that we've done on a particular strain called EC958, and that's part of this dominant clade C cluster. So EC958 is a reference ST131 strain. It was isolated from the urine of an 8-year-old girl with the UTI. It's multidrug resistant, and I won't go into all of the antibiotics it's resistant to, but it has a large plasmid containing resistance genes that mediate this phenotype, and it has the majority of those virulence factors that I pointed out earlier as well. So in order to look at all of its ability to survive in the bloodstream, we devised a particular genetic screen using a method that we'll refer to as TRADIS, or transposon-directed insertion site sequencing. And I'll take you through the method, and I'll take you through then how we've been able to interrogate and identify all of the genes that are required by this organism for survival in serum, and we've called this the serum resistome of E. coli ST131. So here's our method. The first thing we did was we generated a highly saturated transposon mutant library in EC958. So we used a chloramphenicol resistance gene, and this is a mini TN5 transposon, and we combined that with a transposase. And this mixture, the transposon, was electroporated into our EC958 cells, and we were able to obtain a highly saturated transposon mutant library comprising around 1 million mutants. It sounds easy, and I'm not allowed to say it was easy because the person who did the work is in the room here. And I think when we first did that electroporation, there were about 100 colonies on the plate. We were aiming for 1 million, and Yui came into my office and told me the numbers, and I'm scratching my head trying to work out how many electropораtions we would need to do. He managed to optimize that method, and we were able to achieve that high saturation of mutagenesis. This is the experiment that we did, and it's fairly straightforward and simple in its design. We took our library of 1 million mutants, and we incubated those mutants in either fresh human serum or inactivated human serum, with the idea that any mutants that contained a transposon in a gene that was required for survival of this bacteria in human serum would be killed. So we would lose those from the mutant library population, and they wouldn't be killed in inactivated serum. So they were incubated for 1.5 hours in either fresh or inactivated serum. We then took the cells and grew them for 4 hours in LB, and then we centrifuged the cells and extracted the DNA. From this DNA, we then devised a strategy whereby we could amplify the flanking region to the transposon and sequence that. And that was, so we would specifically target this region, and we used Illumina sequencing then to determine all of the insertion sites for the transposon in the population. So we had both an input pool and an output pool. And our input pool is the library as generated, and the output pool is what we recovered after that incubation period in human serum. Okay, so this is some of the data that we got from the experiment. We had around 11 million reads that mapped to the EC958 chromosome, and this corresponded to around 500,000 unique insertion sites. So we estimated that our library contained a transposon insertion approximately once every 10 base pairs. So I think you would agree that that's highly saturated. If we look at the distribution of those insertions, here we have the frequency versus insertion index, which reflects the number of insertion sites per gene length. We can see that that's consistent with that insertion frequency, but also that we have some genes which were very underrepresented in this library. And that means that there were very few insertions in these genes. And the only way to explain that would be that these are essential genes required for growth under those conditions. And so we came up with a criteria, either no insertion, 1 to 5 sites, or greater than 5, but less than, I think it's 10 or 15 or something, based on the significant calculations. And these comprised our essential gene list in EC958. Now, I'm not going to talk too much further on that. I just mentioned to you that among those essential genes that we identified, we've since gone back and looked at all 50 E. coli complete genomes and looked at the prevalence of those genes in those E. colis. And what we found is that here we have genomes versus each of the genes. So all of the white regions here indicate where the gene is identical amongst all of those genomes. So in actual fact, there are 270 conserved E. coli genes that are essential, potentially meaning that there are 270 genes that you could target if you wanted to, or you could use as new targets in the development of new antimicrobials against E. coli. Okay, so let's look at the serum resistome. What we identified in our TRIDIS analysis were 56 genes overall that we predicted, based on this analysis, were required for survival in human serum. So in the interest of time, I'll just go through and take you through the overall strategy. So here's our EC958 genome. There are around 4,800 genes. From those 4,800 genes, 1.2% were identified by TRIDIS to be required for survival in human serum. And I know you can't read those. I'll go into a little bit more detail in just a couple of minutes. Of those 56 genes, we went and did some site-specific mutagenesis. And when I say we, I mean Kate, who's sitting also over there. And she managed to mutate 54 of those genes in EC958. And when we then went back and tested those for survival in human serum, we found that there were actually 46 out of those genes that possessed the phenotype of enhanced sensitivities. This is an incredibly high hit rate of positive hits from this type of screen. 85% of all of our genes identified actually did play a role in this phenotype. Okay, so if we look at some of the clusters or types of genes we identified, O-antigen biosynthesis, lipid A core biosynthesis, enterobacterial common antigen biosynthesis, some of these were expected. And I'll just take you through what the data looks like with one of these examples, the O-antigen biosynthesis. So this is what the actual data looks like when you plot it. Here's our control, or our input pool, and you can see the numbers of insertions plotted there. And here's a group of genes that were required for survival in human serum, and you can see the difference in the number of mutants that are recovered from those two populations. If you look at the LPS profiling, so here we're looking at the O-antigen chain based on SDS page analysis, and you can see based on these profiles that there are differences in O-antigen in each of these mutants. And when this grading here refers to a serum sensitivity scale. So the higher the number, the more sensitive the mutant is to killing in human serum. Now you can see that there are a couple of other genes that also fit this profile of a reduced number of mutants recovered in the test. You can see in this light blue here there is no difference between these two. So these are genes that were not required, obviously, for survival in human serum. And then we see genes like this coloured in grey here, where there are very few hits in both the control and the test, and these are part of those essential genes that I indicated before, because they're not even present in the input population. Now, what else did we find? Well, we found that there were around 45% of the genes that are required for resistance to human serum independent of altered LPS. So I've indicated some of those here, and if we have a look at those, we can see that the LPS profile is unaltered. If I just point out a couple of these genes, one is LPP, which encodes for a major lipoprotein of E. coli. And in fact, this was the most sensitive mutant that we identified amongst all of the mutants that we made with respect to killing in the presence of human serum. Another mutant, another gene, BAM-B, is required for or is a gene involved in beta barrel assembly within the outer membrane. So it's perhaps not surprising that we identified that gene as well. We also identified several novel O-antigen chain length regulators. I've pointed those out here, and this is what they look like when we looked at the actual data. Both of these genes actually cause a change in the distribution of the LPS or O-antigen profiling of the mutants with respect to the wild type. And in actual fact, one of these genes is a regulator that's been associated with intramacrophage survival, so we believe the two phenotypes make a lot of sense. Now, no method is perfect, and when we looked at our data, we observed that OMP-A had been shown in the literature to be required for survival in human serum, but we didn't pick that up in our screen. So we estimated that OMP-A mutants should be present at around 0.02% of the population in our TRIDIS experiment, and we came up with the hypothesis that maybe the release of OMP-A from all of the rest of the cells, and remember this is a mixed population, well, the release of OMP-A from the rest of the cells might provide this protein in trans to complement OMP-A mutants. And in fact, we were able to show that in a mixed competitive experiment, whereby we mixed different ratios of an OMP-A mutant and a wild-type strain and incubated them in human serum. And we found that at low concentrations of the mutant, it was resistant to the killing of human serum. But then as we increased the concentration of the mutant to above 15%, then what we found is that that mutant then became sensitive to killing by human serum. Okay, so I guess what we then predicted was that there may be other phenotypes associated with these mutants that we had identified. As I said to you, we have 46 out of the 54 that we were able to demonstrate the phenotype with respect to sensitivity to human serum. We also predicted that many of the mutants that we obtained might be altered in outer membrane integrity, and we tested this by looking at their sensitivity to SDS, and we found that half of these mutants were sensitive to SDS, and I've indicated some of those mutants in red on the outer of this diagram. We also predicted that some of these mutants might be more sensitive to changes in osmolarity, and in actual fact we're able to identify some of these mutants. So confirm some of these phenotypes. So overall, what you can see in this assay is that we've been able to identify all of the genes required for survival in a particular phenotype in one single experiment. In the past, the only other way you could do this was to make transposon mutant libraries, get a lot of toothpicks, and pick and patch and try and identify these genes, and you'd never really know whether or not you had captured all of the genes required for the particular phenotype. Okay, so I just want to go and switch back now to another type of virulence factor of UPEC and tell you about some work that we've done on a particular type of fimbrial adhesion that's required for colonisation of the urinary tract, and that's a particular type of fimbri called type 1 fimbri. And remember I said to you that these adhesins are critical for UPEC to colonise the urinary tract, because if they don't stick, then they're going to get washed out by the flushing action of urine. So the fimbri that we work on is a fimbri, as I said, called type 1 fimbri. So these are the type 1 fimbri expressed by UPEC here. You can see that there's these long, hairy structures that stick out from the surface of the cell. And in this cartoon structure, you can see how they are configured. And they comprise a major subunit protein, FimH, which makes up the bulk of the organelle. And then there's a fibrillum structure containing an adhesion at the tip called FimH. So essentially it's like the bacteria having an arm with a hand reaching out, and that hand is the adhesion FimH, and that mediates binding to a receptor containing D mannose, and that enables the bacteria to attach to the bladder epithelium in a lock and key interaction. So what's important is that you remember FimH as the adhesion, and you need to remember that it binds to D mannose containing glycoproteins in the bladder. Okay, so these fimbri have been well studied in other E. coli, and there's the genetic organisation of these genes. What we found in EC958 is that there's an insertion sequence disrupting one of the regulatory genes that control type 1 fimbri expression in EC958. First thing we thought was, oh no, we picked the wrong strain out of our collection. It turns out, however, that pretty much all of the strains in that clade C that I pointed out to you as the dominant clade of ST131, they all contain this insertion in FimB. So in fact, we're working with a highly representative strain. We've been looking at some of the phenotypes of type 1 fimbri in EC958 with the view that that insertion means that the regulation of these genes are quite different in this clone compared to other UPEC strains. And we have good evidence for that. I just don't have time to go into the details of it. What we have done is we've developed a genetic system to be able to mutate genes in EC958. And again, keep in mind, this is not trivial because this is a multidrug resistant strain. So we've been able to mutate the genes for type 1 fimbri, and that's indicated here by this bald cell as opposed to this hairy guy here. Now, when we grow these two strains in static media, we observe that the fimbriated bacteria forms a biofilm at the air-liquid interface of the culture. We can also look at biofilm formation in a microtiter plate system, and this is two different types of media with a wild-type strain versus a mutant strain, and you can see that the wild-type strain adheres to the surface of the microtiter plate and forms a biofilm, as opposed to the mutant strain. And we also have a system to study dynamic biofilm formation in a flow cell. And basically what this means is that you have a flow of media through the entire system, and if the bacteria don't attach and form a biofilm, then they're washed out of this system. And under these conditions, if we examine bacteria adhering to the surface of this flow cell, you can see that the wild-type fimbriated strain makes a very strong biofilm, whereas the type 1 fimbri mutant doesn't. We also have a mouse infection model to study colonisation of the bladder. And in this model, we use female mice, and we inoculate these mice directly with our bacteria in the bladder using a sterile catheter. And we can examine the bacteria that are recovered from the bladder at different time points post-infection by using colony counts. If we take our wild type and our mutant strain, we can see that the type 1 fimbri mutant strain is attenuated around 100-fold for colonisation of the mouse bladder in this system. Okay, so in the literature, it's been well described that when UPEC colonise the mouse bladder, they go through a pathway that involves intracellular biofilm formation. So I'll just take you through a cartoon describing that. So the UPEC cells or bacteria bind to the surface of the epithelium in the bladder by way of type 1 fimbri. They then invade bladder epithelial cells and they begin to replicate in these epithelial cells to form what has been defined as intracellular biofilms. And it's believed that these intracellular biofilms contribute to persistence of the bacteria in the bladder. We then also know that they can burst out or flux out of these cells and colonise adjacent sites within the bladder. And type 1 fimbri are required for this process. There is some evidence that these intracellular biofilms also occur in humans because this is an example of an electron micrograph or scanning EM, not by us, but showing the formation of these large clusters of UPEC cells within an infected patient or in the urine of an infected patient. Okay, so when we looked for these clusters of cells within mice infected with EC958, we certainly observed that they do form. This is confocal scanning, confocal microscopy performed at 6 hours post-infection of mice. And in green here we have the stained bacteria, so you can see these large clusters of intracellular biofilms that are formed within the bladder epithelial cells at, as I said, 6 hours post-infection. We can quantitate the number of these within an individual bladder. And then at 16 hours post-infection we see this characteristic fluxing out of the epithelial cells by the bacteria, again stained in green here. So it seems like our strain has most of the hallmarks of infection of the bladder that's consistent with other UPEC strains. Now, we need to keep in mind that this is a drug-resistant strain. There are not many alternatives left for treatment of this strain, so, and infections caused by this clone of E. coli. So we teamed up with a group of researchers at Washington University in St. Louis who have developed a novel type of anti-adhesive molecule that can actually prevent colonisation of the mouse bladder by inhibiting the binding mediated by type 1 Schembri. And this is a molecule that actually binds to the FimH adhesion that I've already described to you. So we asked two questions. We said, can these inhibitors of FimH prevent acute cystitis, and can they treat chronic cystitis? So a little bit of information about these inhibitors. These are small molecular weight compounds called mannosides, and as I said, they specifically inhibit the FimH adhesion of type 1 Schembri. They've been optimized for FimH blocking potency, oral bioavailability, and pharmacokinetics. And ZFH04269 is a lead preclinical mannoside that's been used or as a candidate for oral treatment and prevention of recurrent UTI. So as I said, these mannosides bind to that region of FimH that would normally interact with D-mannose containing glycoproteins on the bladder epithelium. Okay, so firstly, we tested to see whether these mannosides can prevent acute UTI caused by EC958. So mice were given a prophylactic treatment of an antibiotic cocktail containing trimethoprim sulfamethoxazole. This is a common antibiotic treatment for UTI. They were given this for 3 days prior to infection. And you can see that when they were colonized, there was no difference in PBS-treated versus antibiotic-treated mice with respect to the number of bacteria recovered from the bladder. However, when we gave these mice one single oral dose of this mannoside 30 minutes prior to infection, we had around 100-fold reduction in the number of bacteria that were recovered from the bladder. What we then wanted to do was to ask the question whether or not this mannoside could treat chronic cystitis. So first of all, we had to establish a chronic cystitis model, and we did that together with our collaborators, and we did that by inoculating mice and following the number of bacteria in the urine over a 2-week period. And what we found is that over this 2-week period, there were around 40% of the mice that were infected that actually maintained a number of bacteria at around 10 to the 7 at all time points that we examined. Okay, so this is the experiment that we did, and again, it's fairly straightforward in its design. We established chronic cystitis by EC958 in mice, and I've showed you that we could do that by monitoring the number of bacteria over a 2-week period. And then we took these mice that had been chronically infected and gave them either one single oral dose of the mannoside, or we gave them PBS. And 6 hours post-treatment, we sacrificed these mice and we enumerated the number of bacteria that were present in the bladder. Okay, so this is a control mice treated with PBS. You can see that they have a median of around 10 to the 7 CFUs per mil. I'm nearly at the end, so if someone wants to make that noise again to make sure everyone wakes up, you won't miss the final bit of data. So this is what happens with one single oral dose of this mannoside. We see around a 1,000-fold reduction in the number of bacteria that are recovered from the bladder of these mice. So we think this is really exciting because remember, we need to think of this in the context of a multidrug resistant strain where there are really limited treatment options available. Okay, so as a summary, I've told you that this clone of E. coli is a dominant multidrug resistant UPEC strain. It's the genomic sequencing that I showed briefly supports the rapid and recent global dispersal of ST131 as a single clone. The strains from clade C are most common. EC958 is a representative strain. We've now completed its genome. I've shown you how we've been able to define the serum resistome of E. coli ST131 using Tract. I've shown you that EC958 expresses type 1 Schembri and it causes acute and chronic UTI. And I've shown you evidence that manos can significantly reduce colonisation of the bladder by this strain. So what's next here is really thinking about what's next for this clone. There is one class of antibiotic that can effectively treat E.coli ST131, and they're carbapenem antibiotics. And it's now been reported in the literature that there are E.coli ST131 strains that have picked up a gene enabling, conferring resistance to carbapenem antibiotics. So that means that we basically have a clone of E.coli that's resistant to all effective antibiotics that can be used in the treatment of UTI that has already demonstrated its capacity to spread globally. So we don't know what's going to happen, but clearly this means that there's an urgent need to understand this pathogen and to come up with new types of treatments. So just there's a lot of people who have been involved in the work, and just to thank those people. Yui's in the audience, and he really masterminded the TRADIS work. Kate's also in the audience, and Kate made all of those mutants. I'm not sure what the record was, but it was about 32 in a week or something like that. Other people who did the work are highlighted in blue. Makrina Totzika was the person who led the work on the FimH manicides, and we did that work together with Scott Hultgren and his team at Washington University in St. Louis. And I should thank Scott Beetson and his group for the work that they've done on the genomics that I presented. So thanks a lot.